Chapter 2, Section 4, LPS, the outer membrane. Now, what we haven't talked about, Section 3, we talked about the gram-positive cell wall, the peptidoglycan, the thick peptidoglycan layers, teichoic acid, lipoteichoic acid, and whatnot. Talked about the altered version that was going to be found in Archaea with the N-acetyl taloserine. I'm saying that wrong and I know it, but look it up anyways. That cell wall. Well, here we're going to be looking at, in this section, we're going to be looking at the gram-negative cell envelope. Okay. Remember, the gram-negative here are going to have an outer membrane because the second lipid bilayer is to counter the effects of having a thin cell wall. This outer membrane is going to contain a polysaccharide referred to as lipopolysaccharide. It'll be referenced by the acronym in many places as LPS, lipopolysaccharidide. These lipopolysaccharides are used by these gram-negative bacteria for surface recognition, importing virulence factors, pulling stuff in, adding strength to the cell. The outer membrane is also going to have porins to aid in moving things across this outer membrane. This outer membrane is not supposed to be quite as impermeable as the cytoplasmic, so you want to get stuff in and out. And you'll notice here this very thin cell wall, really not going to need much in the way or any teichoic acids because there's really not much there. And these things sticking here, these are the lipopolysaccharides. The lipopolysaccharide, LPS, has what is referred to as a core polysaccharide and an O polysaccharide, two regions here. The polysaccharide means that many saccharides, long chains of sugars, branching chains of sugars for some of these. Turns out the core and the O section have an affinity for cations, calcium, magnesium. The calcium and the magnesium will bind and will cross-link different LPS strands, cross-linking them, adding to the structural integrity, the structural strength associated with that outer membrane. But here's the kicker. The core and the O polysaccharide are attached to a part known as the lipid A portion. Lipid portion, remember the lipopolysaccharide. Lipid A is the part that's actually embedded in the outer membrane. The LPS is going to be the primary constituent of this outer membrane. The O and the core, well, they have some biological things that they do for the cells, but it's that lipid A portion that's part of the outer membrane. This one has a really weird side effect, especially if you are a mammal or us humans, specifically a mammal, that gets infected with, has some form of an infection with a gram-negative. That lipid A portion is what is referred to as an endotoxin. Endo means this is actually part of the cell that gets broken off, released into the environment, and the environment just happens to be you. LPS has this nasty habit of binding receptors. Which receptors? All of them. So you'll start getting cross-linking of receptors on cells that shouldn't be linked, shouldn't be cross-linked, shouldn't be activated. And it starts making the cells, the tissues, respond in weird ways. Yeah, turns out, as we're going to see in later chapters, that infections with certain bacteria, the bacteria itself being there, not a problem. Antibiotics will remove it rather quickly. It's the LPS, the endotoxin, that gets left behind as those cells die and they break apart. That's where most of the damage is going to be. So you see the O, the core, multiple sugars, different types, branching chains, but it's this lipid A right here. This is what does the damage. And it's interesting, if you go and you get a shot, if you look at the vial they're taking it out of, you'll see it'll say endotoxin tested. This is what they're testing for. This right here. Why? Because like I said, this gets injected into your body, into your bloodstream, it goes everywhere. It's going to start causing cells all throughout your body to start acting weird, start sending out signals that shouldn't be sent out at the same time. Next thing you know, you have a fever, you're going into shock. You have a cytokine storm that causes your entire body just basically to shut down. People ask me, well, doesn't that mean don't they just have to sterilize? Well, that's the problem. Anything that's sterile means there's no living cells in it. There are no bacterial fungal spores that are viable left in it. If anything that's going to be injected into a human at any point had some living organism in it, gram-positive, gram-negative, whatever, that shed endotoxins. Here, gram-negatives shedding this LPS. The cells will die. You can autoclave, you can UV irradiate, those die. This doesn't. This gets into our body, you're screwed. And you'll think back to that picture. There was the cytoplasmic membrane, thin cell wall, outer membrane. There was that space in between. That space is referred to as the periplasm. Okay? It is a great place to house things. Remember, these bacterial cells have to pump out protons that'll then flow back in to power transport across the cytoplasmic membrane. Great place to pump them. Periplasmic space. Different proteins need to be made, stored. Don't need to be released quite yet into the environment. Make them, put them in the periplasmic space till later.